3, and we studied longhorn beetle diversity in different height in the primary forest of Mosong using pheromone traps. So the longhorn beetles are the insects that belong to the family Cyromycidae, and they are characterized by the presence of long antennae. In some species, the antennae can be twice as their body length. And ecologically, they are a phytophagous group of insects. That is, their food range varies from wood to the herbaceous plant species. And there are 35,000 species of longhorn beetle described so far, and there is speculation that one-third of its average discovery in the tropical areas. The longhorn beetles can be serious pests in different stages of their life, sometimes the larva boring into the wood or the adult feeding on different tissues of their host plants. We studied the pheromone beetles using, we studied the longhorn beetles using the pheromone traps. As you can see, this is our trap. This is called an intercept trap. And we use the pheromones as a lure because it is known that it is the sensitive traps, and it can really act well to attract insects present at very low densities in forest. Certain studies have said that the Cyromid species can really display a very display preference for various regions of the trees, and keeping this in mind, we chose to study that how the abundance of the longhorn beetle vary in canopy and understory, and whether the longhorn beetles have any height preference, and how the predators of the beetle respond to the lure. And the next part of our study, will be continued by Hugu.
Okay, we conducted our study at Mosong, southwest China, and near the boundary with China, between China and Myanmar and Laos. It's a hotspot for biodiversity conservation. Every day we went through the forest, we can hear the boom boom boom, to kill a bird. So many birds there. And we randomly choose 18 trees in an old growth forest, or we call it the primary forest. And it's in 3 rows, so in each row we have 6 trees, and the minimum distance of each 2 trees is 15 meters. So we put traps at 3 heads, it's 1 meter, 5 meter canopy, and we use the big shot. Big shot to put the canopy trees. It's very hard to do, actually. Almost all work were done by rats. We use 6 treatments and 4 compounds and pheromone oil. It's very complicated. So it's just from the previous study, we know it's very effective. So we use this for these 4 compounds and oil. And we also use to control it's ethanol and blank. And to avoid the operating effects, the operation of the traps will change once during our experiments. And the duration of the experiments is 10 days. And we collect all the insects from the traps every 5 days. So we change the operation in the tree 1 to tree 2, then tree 2 to tree 3 next. And then tree 6 back to tree 1. So we're collecting the longhorn beetle in the field. And the next part of the data analysis will be introduced by Nani.
So we analyze our data using statistical software and estimators. So we use the non-parametric length test to compare the abundance of each of species. And we use the generalized linear model to compare